Okay.
So I'm just going to give another minute for others to join Natalie, if that's okay.
Yeah, that's fine.
And
I see my David Gadori joined already, so we can start. Hi, Dave.
Morning, Natalia.
Morning.
I just sent you a notice in the chat that your fan club is on the way.
My fan club is on the way. my ducklings. How's the weather there?
cold.
It's November in Geneva. It's not hailing or snowing at the moment, so I suppose it's good.
It's cool for Florida standards. It was in the 60s this morning.
Sounds good.
It's perfect.
Well, I guess we can get started. I see some others are joining right now. Well, good morning, everyone. Welcome back to those who have been following our IPM seminar series. This is our third one, and we have a special guest today, Dr. Natalia Perez from University of Florida. And before I make an introduction, I just want to go over some very quick items, just to remind you that we will be recording this seminar, and we will be posting the seminar in our YouTube channel, and I'll put the link in the chat box in 1 second. Also, for those in the bottom of your screen, if you want to use the closed caption, there's a closed caption option as well. You can turn it on. And we want to ask you just to please mute yourself during the seminar. So we have about an hour, and Dr. Perez, after she finishes her seminar, will have a Q&A session. And after that, for those who are interested on having just further conversation and meet with Dr. Natalia Perez, we'll have a great and meet until 12:30. So we want to encourage you to stay for a little longer, and we'll keep the channel open as well. Feel free to put your questions in the chat as well, and I'll make sure to read those questions at the end of the presentation as well. Our next and the last seminar is next month, December 9th. It's going to be Dr. Eric Clifton, and he is with Cornell University, and he will be talking about spotted lanternfly and some efforts on biological control agents and other things that they have been exploring to control that invasive in the state. So now, Dr. Natalia Perez, thank you, Natalia, for accepting our invitation. Dr. Natalia Perez is a professor at the University of Florida at the Golf Course Research and Education Center. She has an active applied research and extension program focused primarily on the management of fungal diseases of a strawberry. She is also responsible for the diagnostic clinic serving the strawberry, ornamental, and vegetable industry in the area. Her research and extension programs focus on the development of IPM approaches to reduce the losses to growers in Florida. She has developed the widely adopted web-based strawberry advisory system, which, by the way, Natalia, as you know, we have our newest system, and I think we used your system as part of our strawberry advisory system in the state as well, which provides recommendations for strawberry growers on the need for fungicide applications based on the weather conditions. Her program is internationally known and has attracted students and visiting scientists from many countries. She has authored or co-authored over 150 peer-reviewed articles in scientific journals, as well as technical extension bulletins, and published multiple book chapters. In addition to numerous awards from the University of Florida, she received the American Phytopathological Society William Boatwright and Mabel Ellen Ball Hewitt Award, the APS Lee Hutchins Award for Excellence in Fruit Research, and was recently recognized as an APS Fellow. Natalia, we're delighted and honored to have you here today, and the floor is yours.
All right, thank you very much for the nice introduction, Alejandro, and thank you for the invitation to speak to you today. So I'm going to go ahead and share my screen. Is that a full mode? Is it good?
It's looking
Yeah.
good, Natalia.
Okay. So, and I'm going to turn off my video to make sure that the connection is usually better that way. Okay, so thank you again and good morning. So let's go ahead and get started. I also like to start with some background on strawberry production in Florida. So just some numbers here, and let me put it laser pointer here just a minute. Okay. So we have about 10,000 acres of strawberry production in Florida, and about 95% of that is located in Hillsborough County in West Central Florida. And that's where the Gulf Coast Research and Education Center is located as well. So I'm pretty close to where the growers are. So it's a high value crop. It generates over $300 million a year. But it's also a high cost, a high investment crop. It costs about $30,000 to put $30,000 to put an acre of strawberry in Florida. So and that's an annual investment. We grow as an annual crop, and starting about right now. I know up where you are, you're not thinking much about planting right now, but we just did that in our Florida fields. We just finished planting. Most of the planting goes on in October. We use mostly what we call this bare root green top transplants. And those transplants are planted in beds. They're covered with black plastic mulch. They're fumigated. And because it's still pretty hot when we're planting and we're using black plastic mulch, we use overhead irrigation to help with plant establishing in the beginning of the season for about 10 to 14 days. But after that, all the irrigation is through drip. We have about 2 peak blooms that will give us 2 peak harvest periods during the season. But once the plants start producing, usually mid-November, in a few weeks, we should have some fruit already. We harvest all the way until really when the market crashes. The end of the season is pretty much dictated by market. Whenever California or Mexico start putting a lot of fruit out, that's the end of the crop for us, and growers will kill the crop with a herbicide or fumigant or whatever, and then start over again the following year. So I want to take some time now to talk a little bit more about those transplants. The transplants that we use in Florida fields are not produced in Florida. They come from different states far from Florida, mostly Canada, California, North Carolina, and Canada. The strawberry propagation starts with meristem plants. Those plants, they'll then produce runners or daughter plants, and that's how you build up the numbers. For the first few generations of those plants, they are produced in screen houses or greenhouses. They're tested and certified virus-free. But then after that, those, what we call those foundation plants, they're planted in open fields, as you see in the pictures here. You planted the mother plants, and those mother plants produce runners or daughter plants, and those daughter plants are then harvested and shipped to Florida, or they are capped to be multiplied for another generation. And as you can see, that stage of the propagation is on large open fields, like you see in the pictures. And with that, there is a room for a lot of the diseases that we see in our production fields. The nurseries face pretty much the same one. So most of the diseases we have in Florida are introduced with transplants from those open field nurseries. I have selected the most important and to put it on this slide. So they're mostly fungal diseases. We only have one important bacterial disease, which is angular leaf spot caused by Xanthomonas frugari. I have divided here the ones on the top here, those three fungal diseases in the top. They cause crown rots, so very similar symptoms that basically kill the plants. And those are mainly caused by Phytophthora cactorum, Colletoticum glorisporioides species complex, or Macrophomina fasciolina. And the ones are in the bottom are the foliar and fruit fungal diseases, most important being botrytis, anthracnose, fruit rot caused by Colletoticum acutatum, powder mildew. And now more recently, we have an emerging issue with Pistolotia leaf and fruit rot caused by Neopistolochopsis. I'm mainly going to talk today about those three, botrytis, anthracnose, and powder mildew. But later when we're talking, if there's any questions about any of those other diseases, we do have projects with pretty much all of this. So because the nurseries face pretty much the same diseases our production fields do, they pretty much use the same fungicides our growers do. And first glance, this list might seem extensive, but when you look closely, it's actually not, because a lot of the products towards the bottom are premixes, either with group 7 or with group 11 or group 3. Some of those products like Kinoxifan is very specific for powder mildew, as well as some of the others on this list. So there's really not as many options that it seems. And when you're using those same products over and over for several generations of the plant, there is a risk and that of emerging of resistance, and that's pretty much what we have seen and reported in the past few years. So I just want to go over quickly and with the situation we are now in terms of resistance. Back in 2013, 2014, we first undetected and reported resistance in Colletoticum acutatum to the KOI fungicides. The KOI fungicides used to be the most important fungicide group for control of anthracnose fruit rot. Is the first report of resistance in Colletoticum acutatum in any crop to KOIs. Bruno Forcellini was a student at work at that time, and we reported the resistance was due to the G143A mutation on cytochrome B, and that resistance, that mutation confers cross resistance among the different strobilurin fungicides, and it's a complete resistance whether when you have that mutation you get no control with that group of fungicides anymore. And unfortunately, just a few years later, we were able to confirm that resistance was coming with the transplants from the nurseries. As we collected samples from different nurseries, we reported that many of those nurseries had some level of resistance to KOI fungicides, meaning that when we get transplants in Florida, they are infected and we have resistance to KOI fungicides, that group is no longer effective, and we end up with fields looking like what we see in the picture here. It's basically no control. Moving to a different pathogen, Phytophthora cactorum, that causes a Phytophthora crown rot. Recently, we also reported resistance to Ridomil or mefenoxam. And again, that resistance was linked to two specific nursery sources. which makes our recommendations to growers challenging because whenever growers had plants from those nurseries, Rhydomil was no longer an option for control, and there's really not a lot of other options out there. And then moving to botrytis, which I like to make this analogy as the MRSA of plant pathogens, because that's usually the first one you're going to see resistance to pretty much everything out there. So we have done quite a bit of work with botrytis, reporting resistance to all the different fungicide classes out there, and the situation on this slide is a few years old. We do have now some resistance to fluoxonil as well. It's here at low levels, but we do have some out there, which scientifically has been great for our group. We have published quite a bit on the subject, but it doesn't make it easier for the growers. And surprisingly, even in the case of botrytis, we have been able to link some of the resistance we're seeing with nursery transplants. And I say surprisingly because we wouldn't think that nurseries would be spraying for botrytis, but the reality is that they're spraying the same fungicides. Even if it's not for botrytis, they might be spraying KYs or SDHI fungicides for control of powder mildew or some other diseases that are important in nurseries. So this was work by one of my students, Michelle. We collected isolates from nurseries in California, in Canada, in North Carolina, and all of those had some resistance to mostly at least 3 different fungicide groups, but some had red isolates that were resistant to up to 6 fungicide groups. That's what the different numbers here mean. There's multiple fungicide resistance, meaning that those isolates are not only resistant to one of those classes, but they're resistant to multiple classes of fungicide. So that brings us to what has been the major goal of my program in the past few years, and we have had many different projects funded from different agencies, but mostly with the goal of integrating different strategies for management across the nursery and fruit production fields for all those different diseases that we have out there. And the idea that we have a chance in Florida, and when growing not only in Florida, but when growing an annual crop, that we can reset and start over every year. And if we could just start with cleaner plants, it would give our growers a much better chance to control those diseases. So there's mainly a few different areas that we can group our projects that we've been working over those years, but all of them with a goal of breaking those epidemic cycles while also reducing the number of and particularly of single-site fungicides, considering all the issues we have with resistance. So we have been looking quite a bit, and we always had trying to identify new chemicals and or biological products for nursery use only. And because we can't just tell nurseries you can use the same fungicides the growers are using and not give them an option. So the idea here is that we'll try to identify maybe some different fungicide classes, some different biologicals, or some different strategies that the nurseries can use. And I'm not going to talk much about that during my presentation today. I'm mainly going to talk about the use of the strawberry advisory system as a means to reduce the use of single-site fungicides. And also I'm going to talk about some of our work in the development of non-chemical management alternatives. So, oh, sorry, I went backwards. So starting with the strawberry advisory system, not sure if this group is familiar, so I'm just going to give a little background. The first prototype of the system was released back in 2000. 2010. So it's been out there for a few years already, and the face has changed a little bit over the years. This is the look of the newest version. So basically what you see when you open the system, a map of Florida with different weather stations that we have monitoring for two of the main fruit rot diseases that we have, anthracnose fruit rot and botrytis fruit rot. So that's the A and B that you see when you open the map and each one of those locations. So as an example, for this one location in Dover, at the time this image was taken, there is a moderate risk for anthracnose and a low risk for botrytis. And the red in the map, which don't have any station right now, it's when we have a high risk for one of these diseases. So you can also see here charts showing how the risk has behaved over the past month. So we have had a few high alerts up to now. This line here is where we are today. We are forecast to get some rain pretty much the whole day tomorrow. So you can see, like, after the line, the forecast is showing that we'll likely have a high risk for disease tomorrow. So growers can look ahead and start planning for spraying today ahead of the risk that's coming tomorrow. So the system was developed by using basically disease infection models that were originally published by Dr. Larry Mellon's group. And what my group did was basically to develop or determine action thresholds for timing fungicide applications according to those disease models. So that work was published several years ago. But what I want to point out here is that in both cases, for botrytis and anthracnose, those disease infection models take in consideration two main parameters. One is the length of the most recent wetness period and the average temperature during that wetness period. And in both cases, we assume the inoculum is present, which is conservative, not always the case. With anthracnose in particular, we have sometimes transplants that are coming infected from one nursery or one region and not another, but we do want to be more conservative. So basically, what the system does is we monitor in each one of those stations as soon as leaf wetness period starts. And here, as an example, where you see the gray shaded area, leaf wetness started about 10:15 at night. So we'll start recording, and then at that time we'll start recording temperature as well. We run the infection models for botrytis and anthracnose, and then as soon as the disease risk crosses the threshold we have determined, it sends email or text message alerts to the growers, and then the color in the web system will change to yellow or red, depending on the risk and depending on the disease. So, I have to say the part that the growers really like is those timely alerts, that they don't have to be looking at the system all the time. They'll get an alert that's very simple. It just says there is a risk and for what station. And then if they want to see more or get a more specific recommendation for what fungicide they want to use, they can go into the web system and get a more specific recommendation. And that's something we didn't have initially in the system. It's something we incorporate later, because what we learned is that growers were not always making the best choice on what fungicide to use when they had alerts for each one of those diseases. So we decided to incorporate more specific fungicide recommendations. So they would be using the best or most effective materials at those times. So the overall goal of the Strawberry Advisory System is really not to provide a better control, it is to reduce the number of single-site fungicides, and particularly during those times where we have good weather. And we are growing strawberries in Florida, which is usually warm and humid, but during the winter, we do have several weeks where we can have cool and dry weather. And this is an example here on the screenshot where from late January until late February, we went a good 3 weeks where there's low risk for botrytis. So there's really not much of a point on spraying during this time. And traditionally, the standard for growers would be spray every week, no matter what. So by following the strawberry advisory system, they can skip that week and then spray when conditions are favorable for disease. And the truth is that even our best fungicides is not as good as like those 3 weeks of good weather. And the data from our trials clearly show that. And this is one example here on the graph below, where we are monitoring for botrytis incidence from about that same time period that we have in the graph above. When we have 3 weeks of good weather, we barely had any botrytis, and for Florida, like 5% of botrytis is really nothing. Whenever we got conditions that were favorable, a few days later, we had up to 25% incidence in our control, and our best fungicide treatment in that trial, which was Canja, only reduced that to about half, and a lot of the other treatments and that trial didn't reduce as much. So not that we can control the weather, but it's just to illustrate how we really can skip those weeks of good weather. So because we do have those recommendations in the system of what fungicide to use, we have to continue monitoring for efficacy and monitoring for resistance so we can adjust those recommendations accordingly. And here I'm showing an example of fungicides we used to recommend for botrytis were those 3 isofetamido, Canja, fluopyram, Luna, and fluoxonil switch. But with fluopyram, we are seeing over the years an increase in the frequency of resistant isolates to a point in the 2018-19 season that close to 70% of those isolates were resistant. So those products were that product was no longer effective and is no longer recommended. So I'm happy to say that the system is being successful as it's been well adopted by our growers. Most of them now follow the recommendations from the system, and I get a lot of questions about what we did to convince growers to adopt. And I like to show this work that is summarized on this one publication as the reason for this successful adoption. And basically we work very close with growers. We did a lot of trials in commercial farms where we would ask growers to in one of the areas just spray when they got the alerts and compare to their calendar weekly sprays. And we're basically able to show them there is no difference in disease in anthracnose, botrytis, or yield. The main difference was they were spraying a lot less if they were following the strawberry advisory system. So the system has been now expanded to many other production areas, including some of the areas that have where the nurseries are, like North Carolina and California. Kansas has been as well adopted in some of those areas, but it has been expanded as part of one of the grants we have. So moving now to the area of we've been working on development of non-chemical managing alternatives. One of those alternatives we've been working on is heat treatment, which is not something new. It's something the nurseries used to use in the past, and they mainly follow a protocol that was developed back in the early 90s for control of cyclamen mites, which call for 49C for 7 minutes. But more recently, most of the nurseries were no longer using that, and the reasons they reported for kind of moving away from heat treatment was that it delayed growth, reduced flowering, but mainly that they felt like it was increasing the spread of Xanthomonas in their plant material. So that's where we start working with heat treatment, and I work with Bill Turchek from USDA and that. So we start looking at Xanthomonas as the one that will be the hardest to kill. And sure enough, what you see in this graph is the survival proportion, and the protocol that was used with hot water 49 for 7 minutes, you can see there's 100% survival of Xanthomonas. So the nurseries were right if they were not, if they had Xanthomonas on the plants, and they were not killing Xanthomonas and likely making it worse. So we for Xanthomonas, we really need higher temperatures and longer exposure times. And we decided to focus on those two that I have pointed here, 48 Celsius for 2 hours or 44 Celsius for 4 hours, which, as you can see, doesn't completely eliminate, but it significantly reduces Xanthomonas. And those higher temperatures are not feasible just because it's too harsh for the plants. So looking at those two time temperature combinations that we start working on, we learned they were effective to control Xanthomonas, but there's also a risk of damaging the plant, some mortality. So and we look at different cultivars and the different combinations. So it was effective, but it was some issues with plant damage. So that's when we decided to look at steam as an alternative to hot water treatment. So with one of our USDA funded projects, we were able to build 2 steam units, and those were built by Ole Mariani. He's an engineer and strawberry growers from Norway. So he built us a unit for a nursery in California, and also some small units on GCRC, which it may look familiar to you, are basically growth chambers that never really work as a growth chamber that we converted in a steam chamber. So we basically have been doing a lot of trials over the past few years, looking at the steam units now, not only for Xanthomonas, but for all the other diseases that we have in transplant production. And we kind of settle on 44 Celsius, and we're looking at 2 and 4 hours, because the 48 is kind of borderline on causing damage to the plants. And we also been looking at adding a preheat step at 37 for 1 hour before the heat treatment. And that came as an idea following some previous work showing that this preheat step can induce heat shock proteins, so the plants can take the heat better later on. So, so here's some data for Coleotrochon acutatum, basically looking at the treatment at 44 for 2 or 4 hours with or without the preheat step. So what you can see here, the 2 treatments that were 4 hours at 44 with or without the preheat step were the best treatments in reduced Coleotrochon acutatum colonization, significant better than 2 hours. And Coleotrochon is one of the significant problems that comes with transplants. We also look at botrytis, and in this case, we actually inoculate with a mix of resistant isolates with the idea that we could potentially break the cycle of those resistant strains. And again, the 44 for 4 hours was better than 2 hours. But the pre-heat step didn't really make a difference, just like it was the case for chloritogicon. However, the preheat does make a difference for the plant. The plants do take that heat better with the preheat step. So then we kind of settle on these now as our standard protocol, 37 for 1 hour, followed by 44 for 4 hours. And we tested that for Phytophthora as well, and only looking at our standard protocol with a non-treated. So the heat treatment also reduced mortality of plants inoculated with Phytophthora. So basically we have enough evidence now that shows the steam is a better alternative than hot water. And that's because it causes less damage to the plants. It's effective in reducing a number of pathogens that I just showed, Monas, Sclerotinia, Botrytis, Phytophthora, as well as powder mildew, which I didn't show. The cons is that the treatment does require some specialized units, and one of the challenges is being with large nurseries in California, because they would like to treat the plants in boxes that are full of plants like this, and getting the heat uniform inside that box of plants is being a challenge. And the other con we can say is that it takes a significant amount of time. But we feel that treating the plants on the different steps of the propagation can help to reduce the risk of spreading diseases among nurseries, because one thing I didn't mention earlier is that the nurseries in California, they will provide plant stock to nurseries in Canada as well as North Carolina. So there's a lot of movement of plant materials. So if we could just add a heat treatment step before each one of those generations or cycles, it could help to reduce the spread of pathogens and the risk of epidemics. So now, let me see how much time. Okay, so now the last part of my presentation, I'm going to talk about UV as also an alternative for managing diseases. So UV, it's again nothing new. Those lamps have been out there for over 75 years. They're used in hospitals for water treatment and in many different other places for sterilization of ambient. And the wavelength that's most effective it's around 254, which is within the UVC range. UVB is also effective, but it would take longer period of time than in the UVC range. So the mode of action of UV is that basically damages pathogen DNA, and UV, as you know, we all know, it's out there every day and coming from the sun, and because of that, pathogens have mechanisms to repair damage by UV. So one of the breakthroughs, I would say, that has allowed us to implement UV for field applications is work done in Norway by Dr. Söderbrand and his group, showing that nighttime applications of UV could be more effective because you don't have that mechanism activated to repair the DNA damage. So that said, what pathogens can be controlled with UV? Well, UV is basically a contact treatment. So those pathogens and pests that are more exposed outside of the plants are more likely to be controlled with UV. Powder mildew being one of those, because they're pretty external to the host. They're usually also more severe in greenhouses and protected structures or tunnels. So, and because of that, they have been used in particular in Europe in greenhouses and in some closed environments as a means to control or reduce powder mildew. What nobody had done before is to try to do that in the field, and that's what we been working on in Florida. So it's a bit challenging to take that greenhouse use to the field. It's not like you can have a static equipment in greenhouse and a timer. We had to develop a system where that was powerful enough that we could apply with a tractor drawn unit that was moving at a reasonable speed. So that work was done in collaboration with Dr. Kadori, who was here in the audience, and really one of the experts on UV. That's not my area. I do strawberries, he does UV, but we collaborate well. So as well as the group from the RPI, the Lightning Research Center, Dr. Mark Ray and Dr. Andrew Borman. So, and here's Andrew in this picture. So we basically develop and design and build units that were designed to cover the shape of the strawberry beds. They had arrays of lamps or germicidal UVC lamps. They were powered by a generator, and it's easier to show them to explain what it looks like in the field. So basically in this video to the bottom here, you see looking from the little window behind the tractor and the tractor application at night. So we did several trials now where we start applying after sunset and once or twice a week at different doses, which are really given by different speeds that we move the tractor. So that work has just recently been published in Plant Disease for powder mildew. Rodrigo Onofre was my student who worked with that. So in 3 seasons of data, the first season we didn't have as much powder mildew, but we saw significant reduce in disease with the UV treatment with or without fungicides at the 2 different doses. But this in 2018-19 was the year we had most disease, and our fungicide treatment by itself failed to control powder mildew, likely because of resistance, but the UV treatments with or without fungicides were effective in controlling. So we see that as a good alternative to reduce fungicide use and also to mitigate the risks of failure of control because of fungicide resistance. So since that, we have many other units that have been built and tested on strawberries and other crops. The beds of strawberries in California, for instance, they're larger or wider than the ones we use in Florida. So we have designed and built units that would fit that bed configuration. We have collaborated with a nursery in Nova Scotia that built a unit for nursery use, which the one here on the top. And David and his group, if you want to know a little bit more, they also working with UV for other crops like grapes and apples, and those units look quite a bit different and taller, much taller than our strawberry units. Because the application has to be done at night to be more effective, we have I think my computer just froze. Try again. I was just about to show you the coolest part of the talk, which is our robots for UV application, but why?
And Natalia, we're not longer seeing your screen.
Yeah, so I know my computer just froze, so let me try. Screen sharing failed to start. Okay. Well, I only had it was literally my last slide where I was going to show the robots, and it doesn't want to share now. So basically, let me try one more thing here. Okay. Can you see now?
Yes.
Okay. So so as like I was saying, because the applications are at night, we have partnered with Saga Robotics in a startup in Norway, and to build autonomous robots for those applications. So this is Torvald, the autonomous robot they built for UV applications. So we have done trials in Florida and some other locations as well. And as an alternative to a tractor drawn unit, that you wouldn't need that driver at night. And David, if you want to learn a little bit more, he has some cool Torvalds there that were designed for applications at Grape. So just find David next door if you want to learn more about that. So here's my last slide as a summary of the presentation, and with basically my views for integrated strategies to manage those multiple diseases and fungicide issues. And number one, I think it was and still is for us to create a better linkage between fruit and plant producers than what existed before. And we have really come long ways there to, but can still improve some extension efforts, in particular to nurseries. Our current recommendation for nurseries is that for them to rely more on the multi-site fungicides and less on the single sites as the basis for disease management. For one thing, it's a non-fruit crop, so it makes more sense for them to have that high level of residue than for the fruit production fields. And at the same time, we're looking for other groups, other options that they could use, and save the single site materials that have lower residue, but also more prone for resistance to the fruit growers. And then we also, because there's really not a lot of new chemistry and different, and it takes time to develop those chemistries and biologicals, that we integrate some of those non-chemical alternatives like heat or UV, and of course resistant cultivars as a way to mitigate those disease issues. So with that, I'd like to thank you all. Sorry about the technical problems at the end. I'd like to acknowledge the funding agents, USDA and FSREF, have provided a lot of the funding for what we do. And here is a picture of my group at some point during the past few years. It changes quite a bit every year. Thank you. Yeah, I'll stop
Thank you,
sharing now.
so much. That was a great information. Thank you. Well, now we're opening the microphone for some questions. So please raise your hand or put an asterisk in the chat box. And I see that Brian, you have a question.
That was just my applause, but I can, yeah, Natalia, I was interested in the heat treatment. And when I think of heat treatment and strawberries, I think of solarization and using clear plastic to superheat the soil. Wondering if doing so before planting would suffice for destroying the Xanthomonas or Botrytis populations for those transplants, or if that additional step is still required when using solarization.
Yeah, so solarization would could be looked as an alternative to manage soil-borne pathogens, and the main one that we for us in Florida would be macropomena that causes charcoal rot. out of the other diseases that we have, like Xanthomonas or even Phytophthora, it comes with the transplants. When we sample the soil, it's not there. It's really coming with the transplants. The one that it really survives from season to season is Macrophomina. We have looked at solarization to some extent, but it hasn't been very successful. I think mainly because the time of the year we have to do solarization during the summer, and it's pretty hot in Florida, but it also rains a lot. So it's hard to reach the temperature for the times that we need, and especially to get a pathogen that is very heat resistant like Macrophomina. So we were not very successful with solarization for Macrophomina. And in the case of the other diseases, like I said, we really need to treat the plants because it's not surviving in the soil, it's coming with the plants.
Great, thank you.
You're welcome. I should also have mentioned that I don't envision the heat treatment as something our growers would do before they planted here, because it can potentially slow down flowering a little bit. It doesn't reduce yield. Eventually the plants catch up on yield, but for Florida, the early season production is very important because that's when the price is better. So you don't want to delay flower, not even a week. I see that as more as a way for nurseries to mitigate that movement of infected material. And I didn't talk about much more or at all about this emergent disease we have now with Vesicular chops. As we know, is like a few nurseries that have it. There's been some movement of plant material, and now it's showing up in other nurseries. So the idea is that if the nurseries, as they get plant stock from another nursery, to treat it before they plant in their fields.
See, Brian Eschenauer.
Yeah. So this is interesting, and I was just wondering, is there much communication with the plant suppliers, the nurseries that are producing the plants for your?
It's much better than what it was a few years ago. One of the SCR projects I got a few years ago was basically trying to get integrated and make them communicate better, because in the past it used to be like the grower got the plants here, they had no idea what that nursery did or apply or what diseases they have. There's not a lot of communication or transparency. It's much better now than it was, partially because we start like doing workshops where we would bring nurseries from all those different regions and our growers and had them in the same room and give presentations to both. So it's much better than what it was, to the point that now this season, just before we got the plant, some nurseries were calling and reporting they have disease, giving the growers option, do you want those plants or feel free to look somewhere else. So it's much better than what it was, but still the options for management for the nurseries are still pretty much the same that our growers have here. And another problem that I learned is that a lot of those regions in California or North Carolina or Canada, they really don't have extension efforts for nurseries. In North Carolina has a really good extension, but it's more for fruit production growers, and the nurseries were kind of left by themselves. So it's much better than what it was, like I said, but there's still a long way to go, I think.
Julie.
Hi, Natalia.
Hi, Juliet.
How are you doing?
Good, and you?
Good, good. So I was wondering, you may not know the answer to this, but I'm curious, what is the northern range of Macrophomina? I'm assuming it might be in North Carolina, but I'm not sure if it would survive up here in the Northeast.
Yeah, that's a good question, and I don't know. I haven't even seen any reports in North Carolina, so I don't know if it goes all the way there. I know it's been a significant problem for us in Florida and for California. It's pretty significant in California as well, but it's kind of out and in the south. I don't know how far north that would go. Like I said, I never seen a report from North Carolina.
And then just a quick follow-up question. The UV robot, is the vision for that down the line to, in terms of commercialization and making it really part of an IPM program, that custom spray type companies or consulting companies would own those robots and they would travel to treat fields, kind of like pollinators,
Yeah.
perhaps?
Yeah, I think so, and I think actually that's the direction that this company we work with, Saga Robotics, is going. They're building a team right now in California, which has much bigger problem with powder mildew that we have in Florida. So they actually have now, I don't know the number exactly, but I think their goal was to have 15 robots that they would serve different growers. I don't know exactly where they are, but that's how they envision that they would offer as a service to the growers.
Great, thank you.
You're welcome.
Any other questions for Natalia? I do have a question, Natalia, with a UV light. So I think the speed that you said is like 1.4 miles per hour. So is there like a range depending on the pathogen and how much you need to expose that UV light to that pathogen to be effective?
Yeah, so yes, so we look at a 1.3 to 2.4, which would give us 85 to 170, and then we have look as high as 200 Jews, and depending on with the 2.5, we get an exposure of 4 to 6 seconds per plant, or if you're looking at a plant. So it depends. We know that's sufficient for powder mildew, and we also have data now showing that it controls two-spotted spider mites as well at that dose or that speed. I had a student that just defended his master. David was on his committee looking at botrytis. And for botrytis, for instance, we would need higher doses that we have looked at, which then reduces the speed of the application. So, but the ones that I think now it can, with the data we have, can be implemented already is powder mildew and two-spotted spider mites. The eggs, it doesn't kill the adults, but it kills the eggs.
That's interesting. That was going to be my other question. If there was like any arthropods that being just controlled by the UV light. So interesting to hear about the mite control. What about thrips?
Yeah, so that the student that looked at mites, he initially, when he started his project, he was looking at both, but he wasn't seeing much of an effect on thrips, so he ended up just focusing on mites. He was a master student, so I think the thrips, they move away. You have to, again, think of the UV as a contact, and if the thrips are not there when you're applying, then you're not getting much of a control. And I think his feeling was that the thrips were moving away when from the light, or were not exposed. They were hiding somewhere, and the flowers were hiding, so they're not getting exposed.
That's great. Thank you, Natalia.
You're welcome.
I don't see any more raised hands or questions, so and Natalia, it's almost 12:30, but I don't know if you have another 5 minutes and anyone wants to stay for another couple of minutes and have a more casual conversation with Natalia.
Yeah, I can stay.
Thank you. So please welcome to stay. I'm going to stop recording right now.